Okay. Thanks so much. Thanks, Danny and Jill and Metaviver for having us. Thanks to all the patients and all of you for coming. You know, I always say as a researcher, we learn as much from you as you do from us. We take away, stories and devise scientific questions to hopefully answer those stories. So my topic, which I will address also dormancy, is, I'm a neuroscientist and I study brain metastasis, tumors that go to the brain. And the way I got into it is we want to ask the question of, well, what is the brain doing to help tumors enter the brain and grow? Because we always think that tumors, when they leave and they spread somewhere, that they'll take off like wildfire. But we're finding out more that the organ that these tumors go to have as much to do with the growth of that tumor. And we call that the microenvironment, the tumor microenvironment. And so it's sort of, we need, what we're understanding is that sort of there needs to be a perfect storm for a lot of things to happen for that metastatic tumor to grow. So just a little bit, I'm just going to go off of these slides. So just a... That's okay. I know it's like, I know. It's my save from coaching my son's basketball and Little League. It's that reaction. So just a little bit, when we talk about brain tumors, I always say there are two types of brain tumors. Those that start within the tissue of the brain. Those are the types that Ted Kennedy and John McCain passed away from, for example, like glioblastoma. You hear about oligodendrogliomas, astrocytomas. And then there are tumors that spread to the brain, so metastatic tumors. And as you can see, this number from NCI, metastatic brain tumors outnumber normal or primary brain tumors by 20-fold. So as a PhD scientist working, my primary appointment being in neurosurgery, neurosurgeons see more of these and work on these to take out and control than they do of the primary tumors. And so this has become a real necessity for us to go after the biology of this disease and why is it now that the tumors are growing within the brain. So the top 3 tumors that go to the brain, sort of in that order, are lung, breast, and melanoma. And unfortunately, if a patient is diagnosed with a brain metastasis, it's a very poor survival rate of over 1 year. Almost 20% of patients, unfortunately, live more than a year. So you see where that necessity comes for us to advancing the curable and understanding that disease. So Chad actually, I'm glad that he talked about this in the other session. So this is Paget's seed and soil hypothesis. I'm going to just walk you through it, and I want to, at the end, emphasize something you guys can do to help us researchers and the way you could spread the communication. So the way metastasis works, so when a patient gets brain metastasis or any type of metastasis, let's say colon metastasis from breast or prostate, it's not that, and I know the majority of people know in this room, but I want to just voice it out for everyone. It's not that, for example, in brain metastasis, that a patient who's had breast or lung or melanoma, that now their tumor is the John McCain style of tumor. It's actually breast cancer tissue growing within the brain. That's what metastasis means, is that the original cell has left the original organ and is now growing in another place. Okay, and the way that works, there are several things that need to happen. So, for example, let's say this is a primary lung or a breast. For that tumor to end up leaving, it needs to start migrating or traveling out of its original site. And this scientifically means invasion, or molecularly what we call it, it's called EMT, epithelial to mesenchymal transition. What that means is that it goes from a normal cell, or a differentiated or a normal cell, to a more stem or undifferentiated cell. Because those are the cells that normally tend to move around the body more. So during development, our premature cells are just moving around to get to their final spot. And tumor cells, for the most part, need to revert back to their younger, quote-unquote, stage to move around. When that happens, they then can invade the pipeline of the body, the blood vessels. At this point, and a lot of it is, it's not black and white, but a lot of it is terminology and semantics. But at that point, they become what's known as circulating tumor cells, and they're flowing through the blood vessels and lymph nodes. And in order for them, in my example, to get to the brain, they need to cross back into the brain at the blood-brain barrier. So it's a very interesting concept. So a tumor that takes the pipeline of the body and goes up to the brain, it's still in the pipeline of the brain in the blood vessels. So the analogy I like to use is, imagine this building is the brain. The working environment, the hallways, are the blood vessels. So when you walked in this morning into this building, you were in the hallway, you were in the blood vessels of the brain. But you're not in the working environment of the brain. The working environment is this conference room where we're all meeting. That's where our brain cells are, not outside. So a tumor can get into the brain, but it's not in the working environment until it crosses this blood-brain barrier. And this blood-brain barrier is a impenetrable gate. So imagine, like a disease like meningitis. You hear about it, bacterial meningitis. Why is it so bad? The brain wants to keep bad things out. So meningitis is bad because that bacteria has learned a way of crossing this blood-brain barrier. And when it gets in, it causes an infection to the brain. Same concept with brain metastasis, brain tumors that come out. They've learned a way of crossing this impenetrable gateway. Now, the other problem is, and which I'll come into in a little bit, is that drugs are very hard to cross this blood-brain barrier. So chemo, certain immunotherapy. So there's a formula force that we can find potentially a drug, but then the alternative is that, well, we can't, a lot of drugs don't penetrate the brain so much. And as you can see here with tumor cells, not every tumor cell type crosses the blood-brain barrier with the same rate. So for example, lung can travel or cross this within 48 hours. Then if you could see with respect to breast and melanoma, breast sits at this junction for about 2 to 7 days, and then it crosses over on average. Versus melanoma, it could take 2 weeks. And so this intrinsically tells us that these tumors, even though there are metastatic tumors, they can act differently. Or why is it that some tumors stay outside in the blood vessels and take a certain amount of time? So these are all scientific questions that we can ask. And when they land in the brain, it's not that they go. They could either, the right microenvironment doesn't exist, or they go into hibernation, or they start slowly proliferating. And what does that mean when I say they need to acclimate? So imagine, you're an American and you want to move from North Carolina to France for good. In order for you to, have a viable and a flourishing life in Paris, you have to learn the language, to speak, to buy food, to buy a house. And that's what these tumors are doing. They're starting to acclimate. They're starting to learn the language of the brain, for example, in order to grow. And that's that microenvironment. And sometimes it takes a long time, and sometimes it doesn't. And that's what the dormancy period is. These are the questions we're trying to figure out of why is it that some cells go into hibernation versus others don't. So this leads into latency. And this goes into that dormancy period. So why study, one of the things that we focus in lab is breast and melanoma, and also lung to a certain point. You can see that the latency for breast cancer that goes to the brain is the longest, mostly years to decades. And this timeline allows us the opportunity to study the biology and hopefully come up with some therapeutics. Versus lung, it goes everywhere, and the latency is much, much shorter. So just a little bit about types of treatments, and this is very broad, but if a woman or a man is diagnosed with breast cancer, or lung or melanoma, what ends up happening, this is the treatment paradigm. They come in to see their neuro-oncologist, they see the number of metastases. Based on the number of metastases, they go different ways. So if you have one metastasis, could it be surgically removed? If it does, then you go through the neurosurgery route. If it's not, then you do what's called SRS, specific beam radiation. So this is called stereotactic radiosurgery. If you have more than one metastasis, then you can either do surgery or this gamma knife stereotactic focused surgery. If you have more than 2 or 4 metastases, then you get whole brain radiation. And with that comes a lot of different other issues, including cognitive ones. And after that, then at the same time, you might be getting systemic therapies. And if that doesn't work out, then you go into clinical trials. So we really need to understand, like I told you, this contribution of the microenvironment. So if you're from North Carolina and you want to go to Paris, and I'm not saying North Carolina is like cancer, but I'm just using that as an example. Okay, sorry. So I'll say California, okay, or anywhere, okay. I know Beth is from North Carolina. Sorry, she complimented me. I didn't compliment back. But so imagine if the brain is Europe. A lot of what ends up happening and the things we think about is the way we study it is, well, what is the tumor doing to the brain to allow it to grow? What we're studying is the opposite. What is the brain doing to allow the tumor to grow? And so this all comes up with microenvironments, cells, cell interaction. So if you think about it, there's a couple of ways cells can communicate. They can communicate within themselves. This is called autocrine, so self-signaling. So they signal to themselves. Or in a microenvironment where a tumor cell talks to its environment, so to a brain cell, that's called paracrine signaling. So it's crosstalk signaling. And the way cells do that is with molecules, little molecules, that are sent back and forth. And so the antennas on top of the cells, those are called receptors. And so they capture them, and that's how communication occurs. So I want you to start thinking like this when you think about microenvironment and how tumors talk to each other. So how do we know? Starting off brain metastasis, is a really complicated, and so how do we know where to even start off? Well, the approach I used, I did my PhD at UCLA, and I did it in traumatic brain injury and neurodegenerative diseases. And I view brain metastasis as an injury to the brain. It's an insult, right? You have a foreign body coming in and trying to land and colonize it, and it causes injury. We know a lot about injury from the brain through brain injury studies, through traumatic brain injury, through concussion studies, through MS, through Alzheimer's. So I've taken that point of view in doing my research. And so I just want to give you a slide on what is brain injury in a very neuroscience 101. So this is a brain injury cavity. And what ends up happening is that the brain wants to protect itself. It doesn't want that injury to spread. And so what it does is it literally puts a fence along the injury called a scar, a glial scar. And what ends up happening is that the main cells that are involved, the brain cells, are called astrocytes. And I'll go a little bit more into it. But astrocytes set up this fence here. You also get stem cells and progenitor cells, so those very young type neurons coming in for help. The other types of cells that come in are your brain cells, immune cells. So we wanted to see if this also, the same thing happens in brain metastasis. So this is a patient, a 45-year-old woman who had a breast-to-brain metastasis in the back of her brain. And we wanted to see, at this juncture here where the tumor ends and where the brain begins, is that the same concept as brain injury? Do you get this scarring occurring? This is the histology of that patient, and you see the tumor here. This is the edge. This is the adjacent brain region. And what ends up happening is we wanted to see if this area is sort of recapitulating what we see in injury. So this is a staining from that patient, and she was HER2 positive. So the HER2 I don't know if you could see it so well, but the HER2 cells are in red here, tumor cells. Green are those astrocytes that I mentioned to you. I want you to take a look at here from 45-degree angle, from going this way all the way down. The normal brain green cells, the astrocytes, should normally look like this. They're very sparse. In injury, what we see in traumatic brain injury, those astrocytes start dividing and they become bigger. And you could see that the staining here, the green, looks stronger here and there. This is exactly what happens in injury. So this proves to us that one of the facts is that metastasis is recapitulating what happens to brain during normal injury. And the same thing, the other fascinating thing is that you have astrocytes living in with breast cancer cells, which was not known before. The other factor is looking at stem cells, like I mentioned to you in injury. you could see that in the same patient, there are these stem cells that are within the tumor. Same exact concept as brain injury. We see it now in metastasis. So the question became, well, what are these three types of cells doing with each other? I mean, we knew, okay, we have a model that brain metastasis follows the same patterns as injury, but why are they there? And so what we figured out in a number of publications was actually when breast cancer cells first arrive in the brain, those stem cells actually come in and try to stop the growth of these tumor cells. So the brain is trying to protect itself from these invasions. But what the tumor does is that it literally outsmarts the brain. It comes in and it releases a molecule. So remember those molecules I told you about where they crosstalk? It releases a molecule, and this molecule is actually very important in neurobiology. This molecule, during our childhood or infancy, it makes these young cells become these adult cells, astrocytes. So the tumor comes in and releases this and changes these cells into these that was trying to stop it at the beginning. And when it does this, these cells actually release something else that help it grow. So now the tumor has literally hijacked the microenvironment for its own advantage. So it's learned that, hey, these guys are bad. They're trying to make me stop growing. I'm going to change them. I'm going to change them to something that'll help me grow. And so many other studies, including, ones from Pat Stieg, DYU, Isaiah Fidler, who are, past MRS and current MRS members, they've shown the same exact things, that these astrocytes are actually helpful to tumor cells, and they can do everything from protecting tumor cells from chemotherapy, or they can somehow turn on the bad genes in tumor cells. And so the tumor cells are hijacking the normal brain cells for their own advantage. And so the last parts of my talk, what I want to do is look at tumors and brain cells, actual brain cells. What is it going? And this is actually an image from our lab. These are tumor cancer cells. Cancer is the Latin word for crab, and you can see that they really look like crabs. And these are brain cells in the green here. And you can see that there is crosstalk going on here because they're actually touching each other. And so the touching happens and the talk happens right at this juncture. And so there are molecules that go back and forth. So the last parts of my talk, what I want to show you is that when we think about brain tumors, it's actually, the brain is not just one neighborhood. There's many neighborhoods. And so we have to think about them as 5 little neighborhoods. And so within those neighborhoods, there are different molecules and pathways going through. And so we asked the question, well, what are the different neighborhoods like? Do certain tumors like to go to certain regions of neighborhoods? So we had a special study going with basically shows that we looked at 1,000 patients who had brain metastasis from breast, colon, lung, melanoma, and renal. And we asked, well, do certain tumors like to go to different parts of the brain? And what can we learn from that, from patients? And when we found out, and the way we did that is basically we looked at SRS images, so radiosurgery, because when there's a focus knife going through, radiation, the way the radiologists look at it is an XYZ plane. And based on that, we could map out where the tumors are in the brain. And because of that, we learned certain trends now. We learned that basically breast cancer likes to go to the back of the brain more often in patients, versus lung likes to go more towards the back right. Melanoma likes to go to the front right. So now you're starting to ask questions here now. Why is it that certain tumors like to go to certain regions of the brain? The brain is not just one organ, but it's many neighborhoods. And so other questions we can ask from this study is, well, colon cancer doesn't grow in certain parts of the brain. Why is that? Is there a certain molecule in that part of the brain that doesn't let it grow? And how could we take advantage of that for other parts and make drugs out of those? And so I'm going to go through these just because I know we started. I'm trying to stay on time. I want to get to the main crux of the barrier. So we basically ask, well, what happens to, the brain has many neighborhoods, but how do we get things into the brain? Going back to the blood-brain barrier. We think of the blood-brain barrier like this, but it's actually like this. It's about, more than 1 million blood vessels. And so to really cross it is very formidable. And so there are techniques that are coming out that we know that we can bypass the blood-brain barrier. So there are drugs that are coming out that we can give it through our nose because the nose bypasses the blood-brain barrier. And if you ask any drug user, cocaine user, they sniff cocaine up their nose. There's a reason why, because it goes straight to the brain, because it passes that blood-brain barrier. So we're taking advantage of that biology to see if we could give intranasal drug deliveries for brain metastasis. And this is something I just wanted to go through where, clinicaltrials.gov, it's very, we're moving in the right direction, because in 2017, there was only 46 trials on brain metastasis, but last year there was almost double, and this year there's over 100. So we're moving towards the right direction of moving all of this research, but we definitely need more to be done, and we've hit the target, the surface. And so the last thing I want to tell you is that you should think of these tumors as the wolf in sheep's clothing. That's the analogy. These guys come in and they trick the brain or any other organ to think that they're the sheep, but they're really, when they become unmasked, they're the wolf. And it takes many hands, and I know I'm running out of time. I want to thank everybody who's been part of this, but specifically one person who I had the fortune of meeting last year, and it was very short, and I really want to dedicate this to her, was Monica Hill. I met her at our MRS meeting in Princeton last year, and it was very short, but we kept in touch, unfortunately, through emails and talks, and I'm a recipient of through Metavivor for one of our projects through her foundation, and I really, I'm very thankful for the work that she did in such a short amount of time. So thank you.
What you could do is you could look at it here and don't even worry about this.
Oh, okay.
But if you want to move this, you just move the space bar.
Okay. Oh, there you go. Yeah, thanks. All right. So I'm going to talk about metastasis as well. My name is Connor Lynch, and I'm from the Moffitt Cancer Center in Tampa, Florida. And we've got good representation here from Florida today. We're trying to get away from the heat to North Carolina. And I will talk specifically about bone metastasis as it relates to prostate cancer disease progression, but some of the mechanisms that are involved also are applicable to breast cancer as well as other malignancies that commonly go to the bone, such as kidney cancer, for example, lung cancer as well is another one that goes to the bone quite frequently. Why we're here today is because of metastasis research and trying to understand why cancer cells have a predilection from the primary site to go to different parts of the body, or why in some cases, such as prostate cancer, it almost always tends to be bone metastasis. So 90% of the patients that die from prostate cancer will have evidence of bone disease in their skeleton. And so this is basically obviously responsible for the demise of the patient. You don't die from primary breast cancer or primary prostate cancer, you die from metastasis. And it is responsible for the deaths of these prostate cancer patients. And as I said, up to 90% of them will have evidence of the lesions in their bones. So, So why do we study this, right? So obviously it's an extremely painful disease. For some of you that have bone metastases, you know that these can be extremely painful lesions that greatly impact the quality of your life and can be very difficult to deal with as well, impacting your mobility. And the current treatment paradigms for prostate cancer specifically are highlighted here on the left. Debulking or surgical intervention actually is quite rare for bone metastatic prostate cancer patients, and the reason being is that these lesions tend to be more bone forming. So breast cancer in bone typically tends to destroy the bone and the bones weaken, and often pathological fracture happens. But the orthopedic surgeon can go in and do limb salvage surgery and replace a certain part of the bone, again to improve the function of that limb and improve the ultimate quality of life for the patient. But for men with prostate cancer in their spine or in their hip, it's a very bony, bulky lesion, a very dry lesion essentially, And so the orthopedic folks typically don't tend to do much work on them. So these men really are reliant on androgen deprivation therapy, ADT, so hormonal therapy, to block the function of the androgen, because even in the bone, these prostate cancers are still very much driven by androgens. So the men that had primary prostate cancer, for example, will have a hormone ablation therapy where they'll be chemically castrated, and that essentially causes the regression of the prostate cancer, much like ER starvation of breast cancer. But the prostate cancer, it rewires itself or evolves to really thrive on very low levels of androgens. So, and androgens can be made not just by the testes, but also by fat cells, for example. Or they can engineer biochemical pathways that allow these androgens to be made and to essentially feed themselves in very low androgen conditions. So much of the work in the oncology realm is still focused on the development of androgen deprivation therapies that really try to go after these prostate cancers that had developed backdoor mechanisms to make the androgens or testosterone that they're really dependent on their survival for. Chemotherapy is also obviously something that is used, like docetaxel in this setting in the bone as well. And recently, one therapy which has been quite successful for the treatment of men with bone metastatic prostate cancer is radium-223. And so this is an alpha radium particle. So it's injected, but it mimics calcium and it actually sticks to the bone. So if you can imagine in the bone, these cancers are turning the bone over or forcing the degradation of the bone. These chemicals like radium-223 bind to that bone area, and because they're short emitters of radiation, they only kill cancer cells that are within maybe a 10 micrometer radius of them. So they're very specific. You don't get a whole body irradiation, for example, of this alpha particle. It just focuses in on where areas of bone are turning over. And the thing is that, you know, for these patients, they're on the clock, and so a lot of these therapies, you know, they may extend overall survival by 2 to 3 years, but ultimately the patient will come down with these lesions in their skeleton. And so from a research perspective, we're still interested in trying to understand why the prostate cancer cells, or any cancer cell, for example, gets to the bone. And when they get to the bone, what are the mechanisms that they use, as Josh pointed out, to really speak the language of the bone so that they can establish and grow in that environment. And really through identifying the mechanisms or the molecules that these cancer cells use to spread from one site to the other, we feel that we can develop targeted therapies that ultimately will be useful for the treatment of the disease. So again, Josh kind of highlighted the importance of the tumor microenvironment. So many of you here are more educated even than us in terms of cancer genetics and the genetics of cancer, and BRCA1, for example, HER2, RAS, etc. And these are really important drivers, intrinsic drivers of the cancer cell's survival and growth, right? And so rightly so, if you can develop a targeted therapy that stops a growth factor from stimulating the growth of a cancer cell, you might have a viable therapy to target that cancer. But we know for a fact that if we look at a histological section, when you look at that slice down the microscope, 5–10% of it might actually be cancer cells, and the rest of it is what we call the microenvironment or the supporting structure, right? And the cancer cells themselves require that supporting structure. They require angiogenesis, which is the formation of blood vessels, for example, in order to feed them oxygen and grow beyond a certain size. We know this for a fact. Fibroblasts that bring in structural support and generate collagen to support the growth of these primary cancers, and as well as obviously the immune cell microenvironment as well. And many of you are already aware of many of the different exciting therapies that have come down the pipe in terms of targeting the immune cells that infiltrate that primary cancer, or that metastatic cancer for that matter, and essentially to awaken the immune system to the presence of the cancer and to knock it out. The other thing I wanted to kind of focus on a few things that might be a little bit different for you all as well is the involvement of neuronal cells that infiltrate that primary tumor. So, you know, for example, many of you feel pain with respect to these primary cancers or these metastatic lesions, and there's a heavy involvement of neurons inside in this microenvironment of the tumor, and a lot of scientists are actually exploring this particular field. And then there are also non-cellular factors. So we talk about fibroblasts, immune cells, neural cells, etc. But also then the things that are made by the primary tumor microenvironment or metastatic microenvironment, such as pH, metabolic metabolites that are generated by the tumors, and also the matrix that surrounds that tumor as well. So counterintuitively, what people have found is that some primary cancers make a lot of extracellular matrix, and when I say extracellular matrix, I mean like collagen, type 1 collagen, like your skin, but it's very, very stiff, and that stiffness actually, while you think it might encapsulate the tumor and stop it from getting away, it actually causes the evolution of a very aggressive phenotype and can actually increase the metastatic potential of some of these primary tumor cells as well. So matrix stiffness is an area of active research. And this is a busy slide, but I'll give you the quick snapshot. But essentially, about 4 years ago now, there was a really groundbreaking paper showing the role of the autonomic nervous system, the ANS, in driving prostate cancer primary growth, and also then when it gets to the secondary site, the metastatic growth as well. So the two parts of the autonomic nervous system are the sympathetic nervous system and the parasympathetic nervous system. And through elegant studies, we've shown that when these primary tumors get infiltrated with neural cells in the primary site, the prostate in this case, it drives the growth of the actual tumor. But only when the prostate gets to the actual secondary site then does the parasympathetic nervous system take control and also force the growth of these prostate cancer cells in the metastatic environment. And I already alluded to this. I've kind of attached some pictures of leaders in the field. These are by no means the people that are the only ones working in it, but the ones that are known. And Valerie here is doing a bit of a Phantom of the Opera, but Valerie Weaver has been one of the pioneers in this kind of concept of matrix stiffness and in driving tumor progression and metastasis. And again, this is where the primary tumor is essentially secreting enzymes that cause the extracellular matrix to get extremely stiff and to force its invasion and metastasis to secondary sites. So you've already heard about this a few times, and I won't belabor it, but this is kind of our current concept of how cancer cells metastasize from the primary site to the secondary site, and this is describing the hematogenous route or through the blood vessels. So essentially the cancer cells will shed off into the surrounding blood vessels and essentially circulate and spread to different sites of the body. But we know that the lymphatic system is also extremely important in terms of a route of metastasis to secondary sites as well. And another concept that people are very interested at the moment, and we heard it talked about in the last session, is this concept of exosomes that are secreted by the primary tumor. So if you think about the primary tumor, in this case prostate, for example, and it's sitting there, it's shedding off growth factors, cytokines. Some of the prostate cancer cells are dying and releasing their DNA, their RNA, different factors into the actual blood. And one of the things that David Leiden here has shown is that, well, you can't see him, but believe me, he's kind of off to the right there somewhere, is that cancer cells, well, all cells actually, but cancer cells more so, can secrete little bubbles, if you will. And these bubbles that are secreted from the cancer cell contain a lot of DNA, RNA, a lot of metabolites. They contain growth factors, cytokines, and they can actually circulate in the blood and hit different organs in the body. And David had some really seminal papers showing that ever before the cancer cell physically metastasizes to the lung, for example, these little bubbles that the exosomes that the cancer cells are shedding are actually interacting with the cells of the lung and establishing a home for these metastatic cells to actually arrive at in the future. So almost like setting up a house for the arrival of the cancer cells. And it's an active area of research, not only mechanistically trying to understand how these little packets of information that are shed from the cancer cells do this, but also what do they contain in terms of a biomarker that we could potentially use for the aggressiveness of the disease or for response to treatment even as well. So it's a really provocative area of research that a lot of folks are working on at the moment as well. We've heard the seed and soil mentioned quite a bit. I'll actually bring a picture of the guy himself there. This is Sir Stephen Paget from 1889. And so we've known for many years that, that certain seeds, certain cancer cells have a predilection for going to certain sites in the body. For example, in prostate cancer, this seems to be the bone all the time. And this concept kind of fell out of favor in terms of vascular flow. People kind of thought, well, the most common site for metastasis is going to be the first organ that cancer cell hits in terms of, when it's carried by the blood. So that's why we get so much lung metastasis, for example, because it's where your blood kind of gets enriched with oxygen before it goes back out into the body. And so that kind of flow pattern was in vogue for many years, but Josh Fiedler, who's in MD Anderson, he's still there and still contributes, kind of resurrected this back in the 1970s. Again, this kind of idea that there is a genetic predisposition of clones within that primary cancer that will dictate whether it can go to brain, to lung, to liver, to bone. And really, 40 years later, we're really kind of making inroads into understanding what some of those genetic drivers are that are responsible for getting the cancer cells to different sites in the body. And perhaps one of the most preeminent researchers in this area is Joan Massaguet, who's at Memorial Sloan Kettering and has shown over the last 15 to 20 years now, using very elegant mouse models, that he can take a single cancer cell line. So you guys know that for researchers, we use a lot of cancer cell lines in vitro to see whether drugs are working or not. And we think that cancer cell line is very homogeneous, right? It's the same cell over and over again. But even in the Petri dish, we can appreciate now that there are clonal variants even within that cell line that we're growing in a Petri dish. And what his lab showed, Dr. Massaguet's lab showed, is that you can take a breast cancer cell line and you can take a single cell out of that breast cancer cell line and you can show that it has more predisposition to go to bone but not lung or brain, lung but not bone or brain, brain but not lung or bone. And this kind of underscores the idea that there are seeds within that primary cancer that are basically genetically wired to spread to certain sites of the body. And of course, if we can understand the genetics behind that, we might know from the primary tumor where that patient might be at risk in the future as well. So Josh touched a little bit on dormancy, and there are many people that are actually investigating this area. It was a question that we had at the lunch roundtables here. It's a provocative area for sure, and I think Erwin will touch on that in the next talk as well. But essentially, this idea that we know for primary tumors that may often look very innocuous, very small, well circumscribed, good prognosis. But if we look at the blood of those patients, we may actually find circulating tumor cells. And there is evidence for prostate cancer, at least, that there are disseminated tumor cells even in very early Gleason stage cancers. And so the thing is that these cells then remain dormant or asleep in these sites, say for example the bone. And what do we do about them? Why are they asleep? We want to know why they go to sleep, and more importantly, why they awaken. So again, many of you in the audience may know of friends that have had primary breast cancer and have been no evidence of disease for many years, yet maybe 10 years later can come down with a metastatic lesion in their hip or their bone, their leg, I mean. So why is that? What are the things that cause these tumors to awaken? And this is an active area of research. Therapeutically, I think, Erwin, you might talk a little bit about it, what to do with the information that we glean, but certainly we are making inroads using animal models to understanding the mechanisms of dormancy, why these tumors go to sleep, and why they awaken. And this is a busy slide as well, but there are three main areas that we think. We think that these disseminated tumor cells can occupy stem cell niches. So for example, in your bone marrow, you have hematopoietic progenitor cells. These are stem cells that give rise to all the different blood cells in your body, all the different immune cells, right? And so they're located within a certain niche of the bone called the endosteal niche. And what cancer cells that arrive there, prostate cancer cells, can do is, because they can speak the language of the actual hematopoietic progenitor niche, when the stem cell comes out of the niche, these prostate cancer cells can enter into that niche and become like a dormant hematopoietic progenitor stem cell in that niche and sit there until they're mobilized by a future event. So this is stem cell niche dormancy. Immune cells can also drive dormancy by constantly killing back or whacking back these metastatic cells that get to the bone microenvironment or other microenvironments, metastatic microenvironments. And then there's the vascular niche as well, which again is the same kind of concept. So even within the dormancy question, we already know that there are different types of dormancy. It's not just that the cell just kind of lands in the metastatic tissue and just sits there asleep. There are different mechanisms of dormancy as well. And understanding those could offer potentially therapeutic insights as well. So as I said, my lab particularly, we're interested in the bone microenvironment for prostate cancer. The bone is a dynamic organ. I like to tell people this. It's not just so that we can stand up straight or kick plants over. It's pretty dynamic. It actually undergoes change almost completely within 5 to 7 years. So it's constantly on the move, right? Metabolically, within your bone, you've got what's called soft cancellous bone. And this is the bone that regulates the calcium levels in your body, for example. But then you've got hard cortical bone, which essentially allows you support, right? And all it is type 1 collagen. It's the same as your skin, except there's calcium really woven into that bone as it's being made by specialized cells. So it's a really dynamic organ. And the guys that are responsible for bone formation are called osteoclast. These are essentially specialized cells that build that bone. They put calcium essentially into type 1 collagen. And the bone is heavily vascularized, as you can imagine. It's the source of all the blood in your body. So it's got extreme vascularization within the bone marrow. It's called sinusoidal vascularization. Very unlike the blood-brain barrier, which is extremely tight to stop things from getting across it. The blood is a bit more fluid. It's not as tight, and it allows egress and ingress out of the bone much more quickly. It's rich in growth factors, so it's not just extracellular matrix. It's got a ton of growth factors in it, like TGF-beta and insulin-like growth factor. And to the kind of yang of the osteoblast yin, we have osteoclasts, and these are specialized cells then that destroy bone. So for patients that are on, that are postmenopausal, that are at risk for osteoporosis, for patients that have aggressive osteolytic disease, they will go on bisphosphonates like Zometa, or you might get denosumab, which essentially targets the osteoclast and stops that bone destruction from happening. So essentially, I've kind of told you really what the osteoblasts do and how specialized they are, and also the osteoclast, then, that it is the guy that essentially resorbs the bone. And this is one of them in action here. You can see it kind of moves along the bone and just destroys it as it goes. And what cancer does then, again, kind of echoing what Josh said at the end of his talk, is cancer cells can speak the language of the osteoblasts and osteoclasts in the bone microenvironment, the ones that are awakening and actually actively growing, to essentially cause bone destruction or bone formation in the case of breast or prostate cancer, respectively. And this is my last slide, but one of the things that will kind of, that is also interesting to us is this awakening. So we talked about dormancy and why cells go into dormancy, but what happens at the very early part of awakening when these cancer cells are triggered to be awoken by some systemic event? And Sean Zhang, who's in Baylor, has had some really neat papers showing that the actual osteoblasts themselves around that hematopoietic niche that I was talking about a while ago often interact with these dormant cancer cells to promote their awakening. And that's a kind of an active area of research as well, because if we can understand those mechanisms, we can potentially come up with targets to intervene and to stop the outgrowth of those metastatic lesions, in this case in the bone microenvironment. So with that, I'll thank you.
Gonna live with that forever.
If you want, move it. It's disconnected, so if you want, just touch this.
Okay, you gotta actually move this.
Okay, there it goes.
If you want, just look at this. Don't even bother with that.
Yeah, Michael.
Good afternoon, everybody. Again, thanks, Danny and Jill, and to Corey and all the people who came here today. I'm going to talk, again, you've gotten a lot of the background from my talk about breast cancer going to the bone and setting up a dormant state. I'm going to talk a little bit about dormancy and reawakening. But my talk is going to be a little bit more about the basic type of research that's done in my laboratory and others in order to find the new gene drivers that control those processes so that we can possibly drug these things, that we can intervene. So these are the new areas where we're learning the new biologies which are much more relevant. So let me step to the side. I've said this already in several different of the, we can't say round tables, square tables, rectangle tables. One of the large problems that we've had in our field for many, many years is that the vast majority of cancer research dollars still goes to looking to treat primary cancer, which for the most part is treated very, very well. So there may be new chemotherapies or better radiations and so on and so forth, but for the most part they're incremental. And so our push is to try to get more funding and more focus on the mechanisms for us to be able to study and the models that copy aspects of metastasis, because that's what kills patients for the most part. And I think the tide is turning, but the reason why we have to set these assays up is because for many, many years, people thought that metastatic cells were just angry primary tumor cells. And even though there's some truth to that, for the most part, they are totally different. They are brain cells in a different environment, so they're speaking a different language. They're speaking French and now loving croissant, but they have the accent of a breast cancer and some of the genetics so that we know it really was, and biomarkers that we know it was originally a breast cancer. So the idea is that yes, this, the idea of immigration or immigrants is true, but we then have to know exactly the pathways that are particular to those metastatic cells under those conditions in order to attack them. If we try to treat them with drugs made for primary cancer, it's a failing idea. So I'm going to talk, so here you have again the, one of the many descriptions in review about primary tumor cell to metastasis. Again, the spread of the cells can be both through the lymphatic system and the vascular system. The one thing I want to point out here, which is a little bit new compared to what other people talked about, is the process of getting these circulating cells then to eventually get cells that move into areas where they've disseminated into peripheral tissues, so these disseminated tumor cells, and then eventually some macrometastasis growing, which is what you could see clinically. And in the middle here is dormancy, which we'll talk about. How often does this happen? And you can see that from a primary tumor, and there's been experiments showing that anywhere from 0.01% to 0.1%, so very, very few cells just get even to this level of circulating tumor cells. Now, a fraction of those, 0.1 to 0.5%, now survive as disseminated cells to peripheral organs, and they now may still stay dormant. And then the question mark becomes, what percentage of those end up to give you growing metastases, which is what harms the patient? So that's what we're going to talk about a little bit. But for many, many years in the field, there's been an appreciation that one of the biggest biologies is dormancy. And why is that? So I'll give you the anecdote about breast cancer. So many of the cases, including Beth, started with metastatic cancer, but no evidence of a primary. On the other hand, you have loads upon loads of studies for decades of women with very early breast cancer who have had double mastectomies, had local lymph nodes taken out. All the pathology comes back and done. We've got it all, you're fine. No margins, no, all the good stuff. And then you take that group of patients and you put the egg timer on them for 10 to 15 years, and a certain percentage will develop brain, lung, and bone metastases. Why? Because we now appreciate, and I'll show you in the next slide, that the seeds of the metastasis must have happened early. So even though you took out the primary lesion, the breast and the breast tumor, there's no more primary lesion, and yet something must have disseminated very early. So Many papers have come out, very seminal papers, including one by the next president of the Metastasis Research Society, Julio Aguirre-Guiso. Really wonderful papers showing that in breast cancer and then in other cancers, that metastatic dissemination can happen very, very early. So the notion that you've now taken out the primary tumor and that's the end of the story is not the end of the story. We would not be here if that was the end of the story. So how do we understand this metastasis and this dormancy, which can happen for 10, 15, 20 years, but then can come back with a predicted frequency? So now we understand that there could be early dissemination of some tumor cells. There's no question that as the primary tumor itself gets bigger, it's shedding more cells. So you have what we call late dissemination because they're making cells that become circulating and disseminated, no question. And then you just have to put the egg timer on and how long is it going to take. Some macroscopic tumors may start to grow after months, years, or decades. So now we get back to some of the things we talked about before that was presented by Doug and Connor about the notion of these dormancy versus active growth. It depends on which cells it interacts with, where in the microenvironment. So many, many different factors. We're just beginning to understand them. So here's a bunch of cells that happen to be in the bone microenvironment. How do you interact with them? And I'm going to show you that there are two different niches. Again, I'm going over what Connor said before, but I'm going to get to a little bit more explanation. Dormancy supportive niches and growth supportive niches. And we now know at least several of the pathways that have to be turned on and turned off in order for dormancy to stay, the cell to stay asleep, or for the cell to reawaken. So several different things have to happen here, and we'll get into that. Now, here I just want to show you that if you look at breast cancer and you compare sites of metastasis as presentation versus the situation that I said to you a minute ago about those women who had, for example, breasts removed, and men could be, breasts removed early, breast cancer, sorry, with the breast and with the lymph nodes, and they were told, oh, you're fine. But then it reoccurred 10 to 15 years later. If you look at the sites of where the metastasis occurs, they're almost all the same place and very similar percentages. Again, bone is the predominant of all those places. So the notion was mentioned before about trying to get brain scans as a prophylactic indicator, as a suggester that you know, even though you have no symptomology, maybe you want to scan to find out if there's going to be a brain lesion. There's evidence that that's the right way to go because brain still is a major site for reoccurrence, so you want to guard against that. All right, so here's the physiology of what a bone looks like. So beforehand, you got sort of the gestalt of what's happening. Here I'm going to try to show you this in the physiology of the bone. So here's the outside of the bone, if you will, in a simple cartoon, which is the calcified hard part of the bone. And then literally the next part of it inside here is this so-called endosteal niche. Again, very, very enriched in these osteoblasts and osteoclasts, heavy in calcium. There's other cells like we just talked about, these nerve cells. And nerve cells, not just any nerve cells, these are ones, remember this, that are controlled by beta-adrenergic receptor signaling. We'll get to that in a second. All right, there's many different types of nerves. And, other cells that are there. But the main point, I should say, there are quiescent. So non-dividing hematopoietic stem cells. So you have to get this term here, hematopoietic stem cells. So these are cells that, if given the right circumstances and the right factors, they will eventually be able to recapitulate any and all of the cells that you find in the blood. But in this niche, they are quiescent. They're dormant themselves. The last thing I want to mention here is if you see at the bottom here, the oxygen gradient. This has the least amount of oxygen of anywhere in the bone. And that's very significant because that's where there's the least amount of vasculature. And as Connor mentioned before, the area here, the central bone marrow, is basically vascular rich. Here's one of the sinusoids, a cross-section of a blood vessel. So it's very high in oxygen, and there's lots of proliferation of cells going on here. And especially you have now activated proliferating stem cells, which are now making all the white and red blood cells that go into your blood later, all right? So in the endosteal niche, again, rich with osteoblasts, these dormant hematopoietic stem cells, they make certain factors. Those are not going to be on the exam, don't worry. High in calcium, low in oxygen. If a breast cancer cell finds its way into that niche, it will become dormant. This we knew from years and years and years of research, and I'll get into how we recapitulated that in the laboratory. If a breast cancer cell gets into this vascular niche, which has these activated proliferating hematopoietic stem cells, high in oxygen, lots of angiogenesis, the fancy word for new blood vessel formation, now any of those cells in there, those breast cancer cells will be actively proliferating and now giving you a macroscopic bone lesion. That's bad. So again, these are going to be busy slides, but what we did is we sort of adapted a procedure done by a different group where we could, in vitro, in cell culture, we could recapitulate the endosteal and the vascular niche, and I'll show you how they worked. So we threw in a bunch of cell lines, bone marrow fibroblasts, these endothelial cells make blood vessels, and human osteoclasts, so you can get these cell lines. That's the endosteal, and in the vascular niche we had bone marrow stromal cells, which are sort of related to the actively growing hematopoietic stem cells. So the way we did it is a little bit hard to see here. We could buy this 3D collagen biomatrix. These are things actually that are used in emergency places. They're actually put on big, if you have a gigantic cut, they're put on top there so you can have, it helps with the healing. So you could buy this. We would have a sterile hole punch and we stick them into wells. And then we have these cells up here or these cells up here, and we could let them sit in there. And again, as you've seen in some of the pictures before, we could artificially label the breast cancer cell lines with a red fluorescence and so on, whereas the other cells in the background we label with green fluorescence. Maybe people in the back or in the front can see that in the actively growing niche, in the vascular niche, the red cells start to proliferate compared to the green cells. You see more red cells there after 6 days. And if you follow those cells, you see that they proliferate, they grow in number. But if you take the same cells and grow them in the endosteal niche, there's not proliferation, so these guys are, they're dormant. And here are a bunch, this is endosteal niche and this vascular niche should be on the right side. Sorry about that. So here you have a bunch of human breast cancer cell lines, and the guys at the top here, some of them are triple negative, which are more aggressive. So for example, MDA MB231, which is triple negative, BT474, and so on. The main point I want to show you is that if you grow them, remember again, this vascular niche is on the right side, they will grow. Here's plus, 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 meaning they're proliferating. But if you have them in the endosteal niche, negative. They sit there dormant for 60 days in cell culture. Every 4 days, we just have to give them some new media, which is food. And we can take them out of that endosteal niche 60 days later, put them onto tissue culture plates, and they are happy and they grow like clams. So they are truly dormant for all that time. It's very interesting. Very aggressive human breast cancer cells grown in these niches just stay dormant. So this is amazing even to us. So based on that, I'm just going to give you a little bit more data. This will be on the exam. All right, just wanted to know your way. So here's that cell line, 231 we call it, and if it's grown in a 2D, which is just a tissue culture plate, you could see that the white shows that it proliferates a lot compared to the control. If you put it in the 3D niche with, like the vascular niche cells, it grows a little bit slower. But if you put in the endosteal niche, it does not grow. Now, Connor mentioned this researcher at Sloan Kettering, Joan Massagay, who took cells of these 231 cells and now put them into mice and found that some of them metastasized to the brain, to the lung, and to the bone. And then he kept taking the ones from the bone or the brain and then re-injecting them, and got some that habituated, that went there with very high frequency. So this is the bone marrow habituated cell that comes from the 231. And you notice that it actively grows in the endosteal niche. It immediately grows, it doesn't care, right? Because it's been habituated that way. So this is our positive control. And I just want to show you that if we have a reagent to knock down one of the major pathway mediators called P38, that's required to be turned on for cancer cells to be dormant in the endosteal niche, P38. If we turn it off, these same cells grow very, very well in the endosteal niche. All right, so we can show that it's dependent on very specific pathways. All right, so what did we do? So here's now where we get to the basic research. I like to look for genes that control aspects of metastasis, that either induce metastasis or suppress metastasis. This is something that Danny Welch and I have been doing. You know, we've loved to do this, and we were the poor orphans of the genetics field for many, many years, and now I think they've realized that we're now, 2 more minutes. Okay, well, I'm almost finished. I only have an hour left. No, all right. So what we do here is we have these reagents called RNAi or shRNA, and they're sequence specific, meaning each one of them will turn off one gene. So we have collections of these things which will turn off every single gene in the genome, but one gene per cell. So that's how we can do it. And we figure out which genes are essential. So if each color of the dot, there's a different gene that we've turned off. And if we now grow the cells and that clone of genes goes away, we know that that gene was essential for the survival of that cell. So very, very quickly we could find out what genes are essential for that cancer cell survival or growth. So we use these, what we call libraries, I'm going to skip this, libraries of all these different clones to knock down genes one after the other, and we're trying to find out if we knock down gene X, Y, or Z, which ones can make our 231 cells start to grow in the endosteal niche. That means that those genes were suppressing reawakening. Did everybody get that? So we start with the 231 cells. They're dormant in the endosteal niche. We turn off one gene at a time. If we turned off that gene, that clone starts to grow in the endosteal niche. That means that gene was a suppressor of reawakening. All right, so we did that, and we got all these genes. We got, different ones. One of the interesting things, just for the scientists, so we could talk about this later, is we had a whole set of genes that control protein production, and those, we don't understand why that works, but that's the case. So we took one gene at a time. We would look at them. Here's one gene. We can here now use our RNAis, shRNAs, knock it down. And when we knock down this gene, here the clone 65, 66, now they grow in the endosteal niche, whereas the control doesn't. All right, so we have one. We have another one called Wnt3. We knock it down. Now we knock it down here with SH Wnt3 clones. They grow in the endosteal niche right here and right here. So I'll just show you a little bit of data to show you that all this can be done very quickly now with thousands, hundreds of patients. We could now take over 500 breast cancer patients in the Cancer Genome Atlas breast study, and we could look at their relative levels of Wnt3, this level, in primary breast cancer versus metastatic breast cancer. And you could see that it's much lower in metastatic breast cancer. That means actively growing metastases turn off this gene. Exactly what we showed, that if we turn off this gene, it actively grows in the niche. And we then showed that if you show that loss of this gene and loss of a very typical tumor suppressor gene called P53, if you look at both losses in breast cancer, That's the red line, and has a very, very strong p-value to say that's a predictor of worse outcome. If you knock off those two genes, turn off those two genes, you're more likely to get the growing metastasis, which is going to kill the patient. Okay? So those are the games we played. These are some of the pathways that you could look at with Wnt2. This is also stuff with Wnt2. And here's just yet another set of genes where we played the same game, yet another one, and so on. And just to show you that bits and pieces of these data were coming out from other labs. This is again from Julio's lab about one of the genes that we talked about. This, I showed it to you very quickly, BHLHE41. And here's a relative of Wnt3 called Wnt5, also implicated to say that if you turn it off, you get worse prognosis and the metastasis grows much faster. So now that's my conclusions, but I want to get to the point about how this might be treated. Because someone asked me before, that might suggest that in patients that have had metastasis but their disease is stable, that they have dormant cells. And I have to tell you, that is highly, highly likely to be correct. But what we want is that those cells either stay fully dormant in you for many, many years, so we maintain them as dormant. What pathways can we learn by these systems that will ensure they stay dormant? That's possibility number one. But there's a second school of thought that says, no, what we should do is before we treat you with your chemotherapy, let's treat them in a early stage with a drug which will smoke them out, if you will, reawaken those cells. So now they're actively growing and now subject to the chemotherapy you're about to get. All right, so I'm giving you some, examples of drugs. Remember the beta-adrenergic receptor positive nerve cells. So here's a drug that is used all the time for asthma, very typical drug. This might be something that could prevent reawakening, and we're working on this in our laboratory and others are too. And here's an example of a Wnt inhibitor. We talked about Wnt. This is very experimental. That might smoke out the cells, make them all wake up, so that if you do this before your chemotherapy, now you would hit them with the chemotherapy. So those are the types of things that have been being looked at. This is very early stage, but again, this would be the next level of trying to work on people's metastatic disease long-term by attacking the issue of dormancy. So I thank you again for this ability to speak. We'll take some questions now. I went too long.